So this is based on quite a large body of work that's being submitted presently from the lab, and we have been doing unbiased screens, both genetic screens and pharmacologic screens for agents that block CRS but do not block CAR T cell function. So the assays were set up in a somewhat simple way. We had target cells that were labeled with luciferase, so we could track their viability by just doing bioluminescence imaging in plates. We added CAR Ts to a target expressed on these leukemia cells that were bioluminescently labeled. And then we had to throw in a third cell, an activated monocyte or an induced dendritic cell. And this was a cell that's primed by specific growth factors. Those cells are critical because those are the cells that seem to produce all of the IL-6. So if you incubate just the CAR T directed towards, for instance, CD19 with a CD19 positive target cell, you get nice killing of the target cell, but you get no IL-6 production. However, if you put in a myeloid cell, in this case, a monocyte, which has been primed in vitro by 2 days of exposure to various cytokines, those three cells together now, when you throw in the CAR Ts, there's an enormous amount of IL-6 being produced. So that's the assay that we use. So we use that to screen hundreds or thousands of compounds. And we also use this to develop models to actually do genetic screens where we would knock out various pathways that are known to be important in inflammation, and also doing some screens or even CRISPR screens where it's completely unbiased screens. So one of the sets of molecules that seemed to work well to block CRS in that model were JAK inhibitors, which I just mentioned. But the most potent inhibitor of IL-6 production, interestingly enough, was Duvelisib, which is a mixed gamma delta PI3 kinase inhibitor. And when we looked at specific gamma inhibitors and specific delta inhibitors, they had about half the effect of Duvelisib. And so when you combine them together, they had a much more potent effect. So it turns out that the lead candidate, as far as our pharmacologic screen, was Duvelisib, which we know is very important in signaling through various pathways in monocytes and in T cells. But we think the primary effect is directly on monocytes, these primed monocytes. And so in that way, what we're doing primarily is allowing the T cells to kill the targets, but blocking the production of IL-6 by blocking both PI3 gamma and delta in the monocytes. And so that's the basis of those studies. And so you would really want to find a small molecule that blocks CRS but does not interfere with CAR T cell function. So we did look at that. We looked at the ability of Duvelisib to block or to mitigate CAR T cell function. We saw no such effect, meaning that CAR T cell function was intact, but CRS production, the production of IL-6 was markedly diminished. And so this is unlike some other compounds that we also found were very potent inhibitors of CRS, but unfortunately, they also blocked CAR T cell function completely, like Dasatinib. And so it's been known through two other groups and published by Crystal Macklu's group and Michael Hudek's group in Germany that Dasatinib is a very potent reversible inhibitor of CAR T cell function. And it does, in our assay, also block CRS. But unfortunately, when you block CRS, you're also blocking CAR T cell function, which is not the desired endpoint. And so we were excited to see that Duvelisib is the lead candidate in our pharmacologic screens. And we're testing that now in a COVID study here at Washington University, where we're giving Duvelisib to patients who have severe COVID pneumonia to see if we can reduce CRS associated with the pneumonias and see if we can improve outcomes. And also we're proposing a similar study using this in the CAR T cell field, similar to the Itacitinib study, which is being tested now for CRS, for CAR T.